All right, so our next presenter is Kurt Deutsch. He's an associate professor at the University of Massachusetts Medical School and director of the psychobiology program at the Kennedy Shriver Center there. He's trained as a behavioral geneticist and a specialization in medical genetics and dysmorphology, and his work is really heavily focused on trying to develop biological measures that are focused primarily on quantitative clinical measures. You'll hear more about the craniofacial dysmorphology endpoints, which are of great interest in neurodevelopmental disorders. So, Kurt, why don't you come up? Yellow means 5.
Thanks, Rob, and thanks to the Autism Consortium. I have to say, Raza is a hard act to follow. That was just stunning to think that there might be some immediate indicator for a forthcoming seizure. So I'm going to talk about another type of surface symptomatology that is related to brain, we think, development. And in particular, we're going to talk about craniofacial dysmorphology. So it turns out that a number of laboratories, including ours, have found a statistical overrepresentation of craniofacial anomalies, like wide-set eyes and low-set ears. There's a very long list of them. But even though there are some replications for this dysmorphology in autism, there were some problems. And these problems really were twofold. One is there were difficulties rendering the diagnosis objective and reliable along the lines Dr. Ring mentioned. And the other problem was you could add up all of the anomalies and get a full-scale score like you would on a psychology or psychiatry inventory, but you didn't know how to interpret the findings. So what we wanted to do is, first of all, render the diagnosis reliable and objective, and also make the variables biologically interpretable. So I thought what I'd do is give you an overview of our approach. We have 3 different elements here. The arrows indicate association. We have the quantitative diagnostic methods I'll tell you about. We also relate craniofacial and brain dysmorphology. So how could that arise? It turns out that the brain and the face are formed by common primordia, or Anlagen. And they're sculpted at the same time by some of the same forces and some of the same genes. So it's plausible that if there were an untoward event, a gene or environmental factor that affected the brain development, it could also affect the delicate features of the head and face. The final row of association here is relating craniofacial and brain dysmorphology to molecular genetic markers. So we decided to adopt an embryologically-derived approach to understand the craniofacial anomalies. And we start very young. It's hard to imagine, but we all started out like this. This is embryonic day 30, and you can recognize some features already in the embryo. Does anybody want to guess what these are? I hear eyes. Yes, that's right. And then these are the nostrils. This is the future mouth, the stomodium. And you can follow the development of these primordia from early on, embryonic 37 days to 55 days, on to 6 months. So that's a baby on the far right. And you can look at the frontonasal prominence. So that's just what it sounds like. The not only the forehead, but also the nose is the frontonasal prominence. You can trace the development of the maxillary prominence, just like it sounds. Those are the cheeks. And you can also follow the development of the mandibular prominence, or the jaw. So from these measures, we can look at embryologically-derived combinations of anomalies, instead of looking at individual features that are of limited theoretical interest. Occasionally, there are variables like that are of interest. The first one that comes to mind is head circumference, because you probably already know that enlarged head circumference, or macrocephaly, is a frequently observed feature in autism. Depending on what your cutoff point is, maybe 20%. So we are interested in what we can tell about chronology. When did these anomalies arise? The type of alteration, is it a malformation, which is geneticist jargon for a primary error of morphogenesis, versus a deformation resulting from a mechanical constraint in utero. And then the embryonic primordia that you just saw can be easily divided into these three regions and their junctions. So this is the type of approach that we use. And it also is useful because it serves as a data reduction device. So what do I mean about the brain and the face having embryologic linkage? Here we see the very top end of the embryo, and this lip of the anterior neuropore is the primordium from which both brain and face are derived via neural crest cell migration. And this is a cartoon. It's kind of mind-boggling. It's very detailed. It comes from a French lab, from the queen of neural crest cell migration, La Douarin. And what she did was use some techniques to look at animal models of brain and face development. So on the left side, this is the top end that we just saw in the EM image of the anterior neuropore. This shows color-coded regions of different parts of the brain. And then in the second picture here, this is a profile of that same embryo with a maintenance of the same color coding, and this is the key down here, for different types of neural crest. And you see that there's a lawful relationship, so that this orange part, which refers to the diencephalic and the anterior mesencephalic neural crest, migrate so that the area around the eye and also the nasal frontal regions are populated by these cells. So taking this type of approach, you can make some predictions about dysmorphology within certain areas of the face and how they relate to regions of the brain. So what about the second prong of the two-pronged approach, the quantitative methods? A lot of people used a scale that was subjective, and there was difficulty obtaining reliability. So when I was an itinerant graduate student in Toronto, I was introduced by their dean of pediatrics there to the world's expert in measuring the head and face in medicine. a dysmorphologist and plastic surgeon named Leslie Farkas. And under his tutelage, I learned his methods, which are very painstaking. But if you learn from a practitioner, you can establish reliability readily. And I'm going to skip over these first two things. The quantitative traits that we measure use the system. I'll tell you about it in a sec. They have not only this element of objectivity and reliability, but also we have an extensive normative database now, which is now coming online, available as face value for geneticists, surgeons, and the like, to condition on age and gender and ethnicity, so you can get a continuously distributed measure of where an individual lies on the distribution. We have initially used direct measurement, or anthropometry, using a system of landmarks devised by classical systems, and also some added by Dr. Farkas. So the face is here festooned with landmarks that have usually Greek or Latinate derivations, but they're easy to identify. And we have been able to take this direct measurement system and apply it to digital anthropometry using the Farkas methodology. And we do that with something called 3D stereophotogrammetry. This is a contraption that gives you a very resolute image of a child, and it's easy to get high throughput images. The analysis is more painstaking, but you can literally, in a matter of milliseconds, get an image on an autistic child. So this neurotypical little boy here has an image that can be zoomed in. You can inspect every detail, and it's kind of uncanny when you first start working with these images. It's almost like staring at the child face to face. And we can perform measurements of the type you saw on the last image regarding the anthropometric landmarks. And we have a protocol that we have applied to the Simons Foundation SSC, Simons Simplex Collection cohort, and also in Los Angeles with AGREE as part of U24 Genomics and Phenomics Center. And now we're moving on to apply these craniofacial dysmorphology methods to other centers, for instance, one at UC San Diego. Now, where can we go with this? We want to somehow come to grips with the problem of heterogeneity. And everyone bemoans the problem. If you start looking at genetic syndromes that are associated with autism, you see that there's an embarrassing number of, an embarrassment of riches. There's such a long list that you really wonder, how can it possibly be that all of these different conditions with their own genetic underpinnings could converge on a single behavioral phenotype? And this became all the more clear when the Simons Foundation network revealed the fact that there are literally hundreds of single variants that will identify and really pepper the entire genome with possible candidates for investigation. Prior to this, there was a lot of energy put into looking at copy number variants. These are regions of the chromosome that relate to either deletions or duplications, literally deletion of genetic material or a duplication of that material, so that you can compare that genotype, a collection of genotypes really, to phenotypes. And the most prevalent and recurrent copy number variant, as Dr. Fischbach mentioned, is 16p11.2 deletion. There is quite a long list of copy number variants that emerged from the Simon Simplex collection. And I'm going to talk about 3 of those shortly. So, applying the same methodology across these different sites, we've been able to come up with comparable sets of data. We have 2 different types of autism collections, the Simon Simplex and then the Agree Multiplex collection. And also we have now, with Jonathan Sebat, who worked at Cold Spring Harbor, who's now at UC San Diego, a broader collection of neuropsychiatric problems, including autism. But here I'm just going to be addressing autism. So we can look at the type of convergent phenotype by looking at the entire group of autistic individuals and trying to determine if there are any particular patterns that pop out. We have found a number of them. The 2 that have the greatest statistical power are craniofacial malformations, primary errors of morphogenesis, and a remarkably high frequency of frontonasal anomalies. So another type of heterogeneity you might look at is the confirmed genetic subtype. We did a study of Velocardiofacial syndrome, which is a 22q12.1 deletion at the Harvard Partners Center for Genetics and Genomics. And we're able to confirm that all of these individuals had the deletion. There is a caricature that can be drawn for VCFS, and that is a collection of anomalies that you can see here. Narrow eye fissures, underdeveloped wings of the nostril, small ears, narrow mouth, a prominent, and by that I mean wide nasal root, hypertelorism, wide set eyes, and flatness of the cheekbones. But the point of this slide is to see how there is such a range of these anomalies. When you measure them objectively, you see that they do, in individuals, cohere into a recognizable case of velocardiofacial syndrome. But there is variable expression of that phenotype. And another example, this is a worked example with a couple of cases from Jonathan Sebat's series, is here for 1q21.1 duplications. And what I did here is I listed 2 cases. Case 1 is a male, case 2 is a female. And we have here, for these craniofacial anomalies, the actual standardized scores. What we use are z-scores, so that's, again, conditioned on the demographics. And what you can see is there is consistency for most of these anomalies associated with 1q21. Wide cranial base is the width of the skull as measured from outside. This is a classical measurement. Frontal bossing, which is protrusion of the forehead, and I'll show you an example in a sec. Also, there are a number of other features here. Here you see, again, hypertelorism. And then you see macrocephaly, and that's a measure that is huge in this one case, over 2 standard deviations above the mean. However, in the other case, it's pretty much normal. not that far from 0. This is frontal bossing. This case is instructive because you see the obvious asymmetry with this moire pattern. And this is an autistic subject, and you find that there's a relatively high frequency of frontal bossing in the Simon simplex collection. Now, how do we come to grips with the phenotypic heterogeneity? What we've done is we've started with a substantial sample from the Simon simplex collection, 140 probands, and used cluster analysis. That's to cluster subjects by anomalies. So what you want to achieve is, again, an objective way of looking at individuals who resemble each other by these anomalies. And then the other shoe dropping is we wanted to relate that to genetics. So we started by relating this to copy number variants. This is in collaboration with the Simons group, Matt State and Stephan Sanders at Yale and Mike Wigler at Cold Spring Harbor, who replicated each other's findings for these patients. And we performed with Hal Stern, some of you may remember from Boston, he's now at UC Irvine, a special type of cluster analysis that allows you to identify these aggregations of anomalies. This first one is called around the lab cluster B1 with highly inclined narrow palpebral fissures, the eye fissures, and a number of other features. And then when we broke blind and went to the CNV database, we found that this relates to kind of a famous region. This is the Williams-Buren duplication region. There were at least 4 cases of Williams-Buren in the SSC cohort. So this is the long arm of the number 7 chromosome, and this region here is duplicated. And just very briefly, 2 other examples. These are regions that have not been discussed in the autism literature. And also, these are syndromes that were hitherto unknown. So I wouldn't call these syndromes yet, but they're putative syndromes. And this particular one, you can see, has a wide array of anomalies. And this maps to the chromosome 7's long arm closer to the end, to the telomere. And finally, there was a cluster that emerged with 3 features that map to the number 3 chromosome, the long arm, and that's a deletion. And each of these genes within the region would be candidate genes for studying the genotype-phenotype correlation. So in sum, we're using these objective measures. We're using them to test brain-face correlations and other studies, collaborating with MRI labs at Brigham and Women's Hospital and Mass General. And then finally, we are correlating molecular genetic markers with these dysmorphic phenotypes. Thank you.